I don't know how to. Oh, okay. So this talk, we're going to move away from Krabbe. This talk is kind of a new talk for me. It's a little bit the future of newborn screening in my mind. So it's going to be thought-provoking and controversial, and I don't know. Let's see where we go. So, so whole genome and whole exome next generation DNA sequencing seems to be coming to newborn screening, whether you like it or not. I think genomics people, some of them tend to think it will provide a universal way to screen for most conditions, but the biochemical genetics people tend to think we face enormous challenges to carry out, newborn screening by next generation sequencing. In my mind, it's not black and white. I'm going to talk a little bit today about whole exome next generation sequencing uh, for newborn screening of monogenic diseases. Let's go to the next slide. I think one of the biggest fears we have about next DNA sequencing is that we might miss some cases. Okay, so it provides kind of a universal way to look at a lot of diseases, but we might have some false negatives. And I think the most comprehensive study we have on this is the paper a couple of years ago from the California group on the next slide to address the false negative problem. Next slide. So they considered 48 inborn errors of metabolism screened in California, as do all states, by the tandem mass spec method that all states are running for amino acids and acylcarnitines. They analyzed about 1,000 high-quality whole exome sequences, and they had 800 with confirmed in the clinic one of these diseases and 388 false positives. So let's look at the 805 confirmed. They used 131 for validation. That is, they analyzed the DNA with knowledge of the patient's phenotype, the type of the disease they had. And then 674, they tried to see if they could explain the disease that the patient has confirmed in the clinic, explain the disease molecularly by having the sequence of the relevant genes. On the next slide. So they analyzed 78 genes that were associated with these 48 diseases. The biochemistry is well understood. And they looked at 3 analysis pipelines. They looked for mutations that were reported in ClinVar and other databases to be pathogenic. They looked at rare variants that were predicted by computer methods to be protein damaging, and they looked for variation in copy numbers. On the next slide. So let's focus in on these 674 newborns that have clinically confirmed disease. And using these 3 arms of curation, they have 93.7% newborn screening positive by next generation sequencing. That means they have 2 or more variants that would be predicted to be pathogenic or known to be pathogenic. So the false negative rate from this sort of massive study is 6.3%. I think the false positive rate would be less than this, less than 6%, had they also included variants of unknown significance that are not predicted to be protein damaging. If we just take the most conservative approach and just say if you have 2 rare variants of unknown significance, this could be screen positive. Anyways, we think the false negative rate is less than 10%. Okay, let's go to the next slide. So in biochemical newborn screening, right, the kind we do now, we try to achieve 0% false negatives, and we get pretty close to this. So 6% false negatives is not considered very good. But we might tolerate 5 to 10% of false negatives because we can screen for a lot more conditions. And how are we going to keep up with all of these new treatments and expansion of newborn screening panels? Anyways, there's a lot of chatter about this, about the fears of false negatives, but I think we have some estimates. Okay, let's go to the next slide. Now let's turn to the problem that worries us, challenges the most, and what do we do with all these variants of unknown significance? And you heard from Dieter's talk that DNA, even among kids that have low GALC enzyme per Krabbe, the DNA is often inconclusive. Cytosine's much better. The reason it's inconclusive is because we find these variants of unknown significance for which we have no clinical information. Most of these are missense variants where we change one amino acid to another, and we simply do not have computer methods to predict the effect of this amino acid change on protein function. I mean, if people say we have some methods, they simply do not work well enough for good enough for newborn screening, where you have to make a very certain call. For some of these variants, we can pretty much be sure that they're damaging. They might be stop codons or frame shifts, things like that. Not always, but mostly they would be damaging. So the biggest problem we face with next generation sequencing are all the VUs for which we cannot predict by any good methods what the consequences will be. Next slide. So we starting to think about this. You can choose sides and say this is impossible, or you can say, DNA, it's coming. I just want to look at the numbers. I'm a numbers guy. Okay, this is my attempt to look at the numbers. So we started with the largest human genomic variant database in the world, which is the Nomad at the Broad Institute of Harvard and MIT. They've sequenced more humans than any place on the planet. It's about 150,000 alleles for each gene in the human genome. Let's take ARSA as an example. The deficiency of this enzyme causes metachromatic leukodystrophy. If you type ARSA into the Nomad database, you get a list of all the variants. So let's eliminate all the variants that are almost surely not pathogenic. For example, ones that don't change the amino acid, things in the middle of the intron, things at the front end and back end of the gene, which almost never causes disease, not always, but. And let's keep the variants that might be pathogenic, like missense, splice site, frame shifts, and stops. Most of these are non-annotated missense variations. They're VUs. One amino acid change, we don't know what they do, but it could cause the disease, right? So now we rank these by allele frequency. We know how many times they find the variant in the database. And we can eliminate any variant that's too common to cause the disease. For example, let's say you have a variant X that has an allele frequency of 1 in 40 alleles. And if you assume this Hardy-Weinberg equilibrium, you expect about 1 in 1,600 people will have, let's say, MLD. If we, I'm talking about iduronidase now for MLD, but we know the frequency of MLD is about 1 in 40,000. So this variant alone would cause more, I'm sorry, MLD. This variant alone, not MPS 1, would cause more MLD than we know exists, okay? So we can eliminate it from further consideration with high confidence. So now what we do is we just sum up all the frequencies of all the variants that are rare enough that they might be maybes for the disease. And this would simply give you the probability of one of the parents being a carrier for one of these variants, okay? I'm not going to go through the math, but it's very simple. Let's go to the next slide. So we can sort of predict how many babies would be born in New York per year that would have a maybe variant / maybe variant. And therefore somebody that we could not say is screen negative with certainty. Somebody that might be a maybe for this disease. But of course, Nomad does not contain all the variants among humans on the planet, right? It's 150,000 alleles. There's some populations that were never included in the first generation of Nomad, for example, Chinese. But we'll get a better sense of whether we're capturing most variants on the planet when the next version comes out, which will be up to about 750,000. So if the number of variants goes up linearly, like 3 times, we know we have essentially no idea how many variants are on the planet. But if it's starting to converge, I think we'll have a pretty good sense of how many variants are on the planet for each disease. Let's go to the next slide. All right, so what I did here is kind of interesting. I took all the variants in Nomad and I estimated how many babies per year would California get if they screened over baby for MPS 1, MPS 2, Pompe, Krabbe, and LD. Assuming that most of the variants are captured in Nomad. And you can do this, the probability the mother's a carrier times the probability that the father's a carrier. Okay, the math is easy. So let's look at MPS 1. 828 babies per year would be a maybe. A VU and another VU. Okay. Washington, smaller state, 141. USA, 4 million babies, 6,000. MPS 2, 187.32, 1490. You see these numbers, okay? So let's take California. For each disease, we have several hundred maybes per year for one disease. All right, let's go to the next slide. Now, this interesting slide, if we start to annotate some of these rare variants, we can reduce the number of maybes considerably. So, for example, if you look at the MPS 1 curve, with all the variants in Nomad that are rare enough that they're not so common that we can eliminate them from being pathogenic. With all the variants now, we would have several hundred maybes. But if we were to annotate, let's say biochemically by expressing these mutants in human cells and looking at the function, if we were to do this on the 50 most frequent variants, the curve, we would have, 828 maybes would drop to about 50. So if we consider, let's say 100 diseases that we might add to the newborn screening panel, it's not going to be screening for everything. Let's say we have treatment for 100 diseases in the next 10 years. Let's focus on those 100. Those are the high priority for screening. We could annotate the 50 most frequent VUs for each of these 100 diseases in a year or two. I think no problem in a high throughput lab. So we could get to a reasonable number. Let's keep going on the next slide. All right, so let's go MPS 1 in California. 828 maybes per year with current Nomad. And let's say it's a few thousand per year if Nomad captures, 20% of the global variants. I don't think it's off by more than that, but we're not sure, right? Let's say we can get the number down to 50 to 200 if we biochemically annotate the high frequency VUs. Okay, so we need to test, say, a few hundred to a few thousand newborns per year by measuring the iduronidase biochemically in their dried blood spots as part of newborn screening. We're not going to call a few hundred families if only 1 to 2% of them really have MPS 1. That would be a disaster. Okay, I don't think society can handle that. Yeah, now, currently in California, we test 500,000 babies first tier by enzyme activity per year. And we could reduce this to, let's say, a few hundred or a few thousand if we do first tier sequencing. And we might extrapolate this estimate to most monogenetic diseases that we want in our newborn screening panel. Okay, so let's go to the next slide. So it seems certain to me, and I think it's obvious to many people, that if we do first tier sequencing for newborn screening, we need to do a biochemical second tier test for each disease in the panel as part of newborn screening, since most of the sequencing first tier hits will be false positives. But what about diseases for which we have no dried blood spot biochemical second tier test? Turns out, and this might be controversial to you, we think we can develop a biochemical second tier test for most monogenetic diseases. I realize this is a very bold statement, but I don't have time to talk here, but just to give you a few examples, we now have biochemical tests in blood spots for things that were once thought to be unscreenable. For example, Wilson disease, which is a deficient copper transporter in Golgi membranes. We couldn't possibly develop a functional assay for a copper transport. What about primary immunodeficiencies like XLA, DOC8, Wiskott-Aldrich syndrome? Cystinosis, tuberous sclerosis. We now have good biochemical screens for these once thought to be unscreenable conditions. All right, let's go to the next slide. All right, so here's my scan into the future of possible roadmap to handle newborn screening for 100 monogenetic diseases, Genomic. It seems imaginable that every newborn screening lab could carry out whole genome sequencing on DBS for all newborns in their state. This is one technique for all states. Yes, it's too expensive and slow now, but in the near future, I think most people think it's coming. For example, there's talk about doing this on 100,000 newborns in the United Kingdom at the federal level, spearheaded by the equivalent of the National Institute of Health. This is coming, okay? It seems silly for each state lab to set up 100 second-tier biochemical screens. Although many of these can be multiplex, this is far from trivial. Okay, as we all know, Joe will tell you this, Dieter will tell you this. So instead, maybe we set up 5 national labs, each responsible for DBS biochemical second tier for, let's say, 20 of these 100 diseases. And each of these 5 labs receives blood spots from all states. Now, I realize, this is fantasy and may never be legislated, but this is my roadmap, and other people might be thinking this way. So each centralized lab would then have to test for 10,000 to 20,000 dried blood spots for the whole country for 20 diseases per year. And the number will continue to drop over the years as we eliminate more and more VUs that turn out to be benign. So the good news for Mike Gelb, Dieter Matern, and others out there that do biochemical, no matter how much DNA we sequence, there will always, and I mean always, be a need for biochemical assays. I mean always, as best as I can say it. So to all the biochemical genetics labs out there, please don't worry, the DNA people are not going to take your jobs away. And to all the biochemical labs out there, there's nothing you can do to stop the DNA people from coming, I'm afraid. So it's not black and white, and it may even be doable. On the next slide. Just to thank, this was mostly done by me with the help of Madeline Campagna. I just want to say Maddie is the daughter of the nurse who took care of Trevor Aldrian. Most of you know the Aldrian, Steve Aldrian, for 5 years, sorry, not 5 hours, before he was lost to Krabbe disease last year. And I'm working with a team of people, most notably Anna O'Donnell-Luria. She's the lead scientist for NOMAD at the Broad Institute at Harvard, MIT. So, I'm talking to serious genomics people. I'm not a genomics person, but it's not that hard. And I have no funding or conflicts to disclose. Thank you.